So you can do it either way, really. We learned about the probabilities of inheritance. So what is the probability, for example, of getting a sickle cell allele? What is the probability of getting particular traits from your parents or even from your grandparents? So that's what we looked at last term. So this term, we're looking at molecular genetics. Molecular genetics. I remember in university, I loved molecular genetics. It made so much sense. We did we did population genetics first, and I almost failed that course. So yeah, that was the only course I just had such a hard time with for some reason. I'm all the Hardy-Weinberg equilibrium and stuff like that. But one thing I did do was study meiosis until I was blue in the face, and then fortunately that was a big part of the final exam. But over the half the class got less than 50% on the final exam. So it was curved, fortunately. But so molecular genetics was like a breath of fresh air because it is chemistry. Of course, not everything is known, but still a lot is, and it makes sense, and it solves, at least in part, this huge mystery of how proteins are made, the proteins that are the traits of every living thing. We're at protein-based life forms. So this will be the outline. We'll talk about how DNA codes for traits, what is a gene, transcription, translation, and mutations. So that's our outline. The flow of genetic information is such that the content of DNA is in the form of very specific sequences of nucleotides along the DNA strands. C, G, A, T, T, C, T, A, G. And then, of course, we'll make the other strand. It's your strand of DNA. And there, I just made a sequence. So, and there's only those 4 nucleotides, but man, can you get a lot of variety out of those 4 nucleotides because they can be in many different orders, many different sequences, and lengths, of course, as well. So there is an enormous variety to be had from those 4 simple nucleotides. I would imagine it is not unlimited, but practically. So the DNA that's inherited from your parents leads to specific traits, and those traits are dictated by the synthesis of proteins. Proteins make your traits, whether the proteins are structural, whether they're transport proteins, whether they're communication proteins, or whether they're enzymes that make other things that make up your body. That is what you are. So the process by which DNA directs protein synthesis and gene expression includes 2 stages called transcription and translation. Well, how did it all get discovered in the first place? Well, in 1909, Archibald Garrod suggested genes dictate phenotypes through enzymes that catalyze chemical reactions in the cell. So he was a mover and shaker in the gene world. It could have been other things. I mean, some contestants for that job were proteins. They could have been the ones that held the inheritable information. But with experimentation, it was found, no, that it wasn't proteins, it was actually genes and DNA. And Beadle and Tatum, they were the first to develop what's known as the one gene, one enzyme hypothesis. But when you look at things, simple animals like fruit flies that have similar numbers of genes as more complex animals such as humans, there's a reason why you can get more complexity from a similar number of genes And that's because there's a minor revision to the one gene, one enzyme hypothesis, which is that genes code for polypeptide chains and for RNA molecules. And one sequence of DNA codes for more than one polypeptide. I'm going to write that in, for more than one polypeptide chain. What are transcription and translation? Transcription is like transcribing. You know how you transcribe? I had a friend here in the building who was actually a creative writer, author, but he was Czech. And so he translated his mother's books. His mother wrote children's books in Czech, and he translated them into English. So he was a translator. But transcription is when you take a strand of DNA and you synthesize RNA using that strand as a template. And it produces something known as messenger RNA. Translation is the synthesis of a polypeptide, and it occurs under the direction of messenger RNA. And it occurs on a cool structure known as a ribosome or ribosomes. This is two subunits of ribosomes. So interestingly, in prokaryotes, they occur together, transcription and translation. So the moment the DNA is transcribed into RNA, it gets immediately translated into a growing chain of amino acids, which is what proteins are. So that's a prokaryote. They're somewhat different than eukaryotes. In eukaryotes, the RNA transcripts are modified. It's called RNA processing. And that's something that's been unveiled, you know, I don't know, maybe in the last 25 years or so. So the nucleus is a separate place from the cytoplasm, as opposed to a prokaryote where it just all takes place in the cytoplasm. I think one of the reasons it all has to take place awfully quickly too is so that that messenger RNA doesn't get eroded by enzymes in the cell itself before it has a chance to be translated into the protein. So this is the nucleus here, and that's where transcription takes place, and that's where RNA processing takes place. But the messenger RNA, it just beetles on out through a pore and ends up in the cytoplasm. And that is where translation takes place, in the cytoplasm. And that's in a eukaryote. So the mRNA is before it becomes a process called pre-mRNA. It's hard to say, pre-mRNA, before it's processed. And then we just call it mRNA. Messenger RNA is what that stands for. So the code is very interesting. The code is cells are governed by a cellular chain of command. Of course, DNA, messenger RNA, and the protein. And how many bases? What is a base? That's my question to you, to recall. What is a base? Before we can ask how many bases there are, I better recall that part. What part of the nucleotide? So what are the three parts of the nucleotide? Do you remember? That may be important for our
nucleoid, the phosphate.
Yes, so there's the phosphate group. Yeah.
And the nucleoid that is made of I'm having a wonderful brain fart right now.
No, no worries. The nitro the nit the base is a nitrogenous base, so it's a nitrogenous ring or two rings. Yeah, that's part of that nucleotide. No worries. I've been having those all morning. So the, the third part then is the sugar. Yeah. So the bases are the purines or pyrimidines. And how many bases correspond to one amino acid? Turns out to be 3. And they're called triplets, base triplets, or more often we use the term codons. And each 3 base codon corresponds to one amino acid. So during transcription, the gene determines the sequence of bases along the length of the messenger RNA molecule. So here's DNA, the double helix, 3 different genes shown here. And this gene here, here's a part of this gene here, just showing one strand, because only one strand is transcribed. So in a DNA parent molecule, only one strand is transcribed. And here's the messenger RNA molecule. And the letters got a bit wonky here. ACC, UGG, etc. And then one of these is a codon that will translate into one amino acid. In this case, it's tryptophan. TRP, tryptophan. So each codon gets translated into one amino acid. And here's the code, cracking the code. So somebody cracked the code. And remember, this is very important, that the codon that is in the code is messenger RNA. That's what's used in the code. So if you have a code such as UUU, whoops, I put a W, UUU, this is the first base, this is the second base, and this is the third base in that codon. So when you're looking at the code, you look for the first base here, and there's the U that we're looking for. So we know that we're looking at this row. The second base, we're looking for the second base. Here it is here, U. So we know we're looking in this column. So it must be in this box here. Then we look for the third U. This is the third base. Here it is here. We follow that along, that row, and we end up finding the amino acid that corresponds to that codon, phenylalanine. So that's how to read the code. We'll do an exercise involving that later. So they must be read in the correct reading frame in order for that polypeptide to be produced. Of course, the genetic code is nearly universal. It's shared by organisms from very simple bacteria to the most complex animals. Indeed, the genetic code is so universal that in lab experiments, you can take a gene it can be transcribed and translated after being transplanted from one species to another. So this is a goat, for example, that uses a human gene for a milk protein. But the transgenic goat, any animal that has genes from a different species is called a transgenic. And it produces a human milk protein that can be harvested from the goat. So, RNA synthesis. Well, the first part of transcription, because RNA synthesis is what transcription produces, it's catalyzed by polymerase. So there's quite a lot of enzymes involved in different processes of replication, which we did before, which was DNA polymerase. But now we're talking about making an RNA strand. It's called RNA polymerase. Poly meaning many, polymerase. It's an enzyme, and it's an RNA polymerase enzyme that catalyzes it. And this RNA polymerase, it pries the DNA strands apart. So remember in replication, we had an enzyme called helicase that separated the DNA strands. In the case of transcription, it is the RNA polymerase that performs that task. It's not a separate enzyme. And it binds together the RNA nucleotides. It follows exactly the same base pair ruling as DNA, but of course, an RNA uracil substitutes for thymine. So there are some differences, of course, between those processes. So the stages of transcription are 3: initiation, elongation, and termination. How does the whole process start, you might ask? Well, on DNA, this is DNA here, there is a sequence known as a promoter sequence. Whoops, it's supposed to be green, sorry. A promoter sequence. That's the start point. And that promoter sequence is at the start of the gene. And that's called initiation. So this larger molecule here, that's RNA polymerase. And RNA polymerase binds to the promoter. It unwinds the DNA, and the polymerase initiates synthesis at that start point. So the template is the one DNA strand. In this case, it's this strand. And here is the new RNA strand being synthesized. That's the start. That's the start of it all. And then that RNA strand starts to elongate. It gets longer and longer. So here it is here, getting longer. And the polymerase moves downstream, unwinding the DNA, elongating the transcript from the 5', I should say 5' to 3'. Because a new nucleotide can only be added onto that 3' end of that growing strand. And eventually, the RNA, the new RNA transcript is released. So it gets to a point where a signal on the gene, at the end of the gene, will signal the polymerase to disembark at that point. So this is the polymerase, and here are the new nucleotides being added on. So how does that happen? the binding and initiation by promoters. So they are the signaling sequence on the DNA. And how are they recognized as that particular gene? By molecules known as transcription factors. So they're initiating the transcription of that particular gene. So they help that RNA polymerase recognize promoter sequences, the ones that are required by the cell itself. And the promoters often have very distinct kinds of sequences. They're often called a TATA box because the sequence is all just thymine and adenine for the most part. Here are the transcription factors. They've often come into the cell because of signaling from outside the cell. Yeah. So as the polymerase keeps moving along the DNA, it continues to untwist the double helix. It exposes about 10 to 20 bases at a time for pairing with RNA nucleotides. And there is some modification of RNA after transcription. It's quite interesting. The pre-mRNA is modified in quite specific ways. It doesn't happen in prokaryotes. And one of the reasons is that that little messenger RNA has to leave the safety of the nucleus and go into the cytoplasm, and it has to find the ribosomes that are going to translate it, or rather the ribosomes have to find it. So they're modified. The 5' end, this should be a prime there, receives a nucleotide cap. It's a guanine cap. And the 3' end gets a poly A tail. That's a tail of adenines, just a bunch of adenines in a row. They don't code for anything. So one of the hypotheses for the reason for the tail is that possibly enzymes that are in the cytoplasm can come along and start to erode or digest that messenger RNA molecule. And it doesn't matter if they digest the AAAs, but also it could help to end the translation process. So another interesting modification is that there's some splicing, RNA splicing. So this is the messenger RNA here. The 5' end, the G cap there, the guanine cap. And you notice it says exons, introns, exons, introns. So exons will remain, but introns are often spliced out. And when they're spliced out, all that you have left are the exons. So that's just the names of those particular sequences. So that's called RNA splicing or alternative splicing. And it's carried out by these neat RNA enzymes called spliceosomes in some cases. So this is it's a little snRNA, it's called. It's a protein / RNA molecule. Some other proteins get involved and create this large molecule called a spliceosome. And it literally cuts this piece out, So it just gets resorbed. And the remaining messenger RNA here, red please. That's the messenger RNA that will be translated. Very interesting. So a ribozyme is a catalytic RNA molecule. They function like enzymes, they splice RNA, and they're one of the clues that RNA was probably the first nucleic acid as opposed to DNA. And the presence of those introns allows for alternative RNA splicing. So it's not always spliced in exactly the same way. And that's because proteins have sometimes what's a modular kind of architecture. They're not just one unit that does one thing, but they could be a bunch of subunits, each of which have a different function. One function may be to bind to something. One function may be an active site. So these are called domains. So different exons may code for different domains of a protein. Domain 1, 2, 3 here, that have different functions of that single protein. So translation is RNA-directed synthesis of a polypeptide. So now we've got that messenger RNA. It's all been processed. But it doesn't work alone. It works with other RNAs as well. Here's the basic concept. Here's the basic concept. Two large ribosomes. Well, one is larger than the other. It's called the large subunit. And this is a small subunit. And they only get together for translation. Here's our transfer RNAs. They carry the amino acids. And they bring the amino acids and temporarily bind to the messenger RNA. And they can do that because this is called a codon on the messenger RNA. But our beautiful transfer RNAs, sort of the general shape of them, brings along with it a piece, because this is an RNA molecule, with an anticodon, it's called, an anticodon. So that's how the transfer RNA carrying the amino acid knows where on that messenger RNA it should be. These tRNAs, they're not all identical because each one carries a specific amino acid on one end and an anticodon on the other end. That's specific, an anticodon. So here's our RNA molecule. Here's the attachment site for the amino acid on one end. And on the other end is the anticodon. It's kind of a looped molecule. That's it in 2 dimensions, but this is what it might look like, something like that in 3 dimensions. That's why this particular shape is used to portray it. So this is the attachment site for the amino acid and the anticodon at the other end. It's important to have that structure. An enzyme called aminoacyl tRNA synthetase. Remember, if something has synthony, you know it's got to have something to do with synthesis. Joins each amino acid to the correct transfer RNA. So here's the enzyme here. It's a fairly large enzyme. Here's our amino acids. One amino acid. And this takes energy. It takes energy from ATP. So here's our ATP. And it splits, it loses two phosphate groups. And that action joins the amino acid as adenosine monophosphate. And that becomes a little molecule in and of itself. So now the amino acid is joined to the adenosine monophosphate, covalently bonding. So the appropriate transfer RNA, though, here comes our appropriate transfer RNA that we want, covalently bonds to the amino acid and it displaces the adenosine monophosphate. Boom, you're out of here. It's my turn now. And now we get the transfer RNA with the appropriate amino acid attached to it. And it's released from the enzyme. Ribosomes are really important for this process. They facilitate this coupling of transfer RNA anticodons with mRNA codons. And there are two subunits, large and small. This is a computer model of what it may look like, possibly. It's a large subunit, the small subunit. These are all tRNA molecules here, different ones. Here's the messenger RNA. Being read. And our beautiful new protein is exiting through a little tunnel in the ribosomal subunit. So the process is such. Within the ribosomal subunits, there are 3 sites. They're known as the P site, the A site, and the E site. So the A site is where the aminoacyl tRNA binds. That's the tRNA that is carrying the amino acid. The next site, the P site, that's the binding site. That's where the new amino acid gets attached to it, the already forming string of amino acids. Well, once this tRNA that was here gives up its string of amino acids, it exits. So this is another site known as an exit site. So here's the exit site. This tRNA, you'll notice something very conspicuous about it. It doesn't have an amino acid anymore. So it's given up its amino acid. Here's the tRNA with a growing chain of amino acids there. Here's the brand new one that just docked. So this chain of amino acids is going to transfer over to that one, and it will be bound by a peptide bond. And there's 3 stages of translation as well: initiation, elongation, and termination. The initiation stage brings together messenger RNA, the tRNA bearing the very first amino acid, which is always methionine, and the 2 subunits of a ribosome. So that's the initiation stage, bringing everybody together to start everything off. So here's methionine. It takes some energy. In elongation, we've looked at that a little bit already. It also takes some energy. To form the new bonds. And I don't really like this one terribly much. We've already seen it happen, really. Here is a tRNA, a growing polypeptide chain. Here comes a new one, new tRNA. It sets up right beside it, and that's handy. So the growing polypeptide chain gets transferred over to the new one. This one then moves over to the exit. As this one exits, the one that just landed, well, it can move over. how does it terminate? When it reaches a stop codon in the messenger RNA. So the stop codons are UAG, UAA, or UGA. When the ribosome reaches that, then the A site of that ribosome accepts a protein called a release factor instead of tRNA, which is interesting. So a release factor hydrolyzes the bond between the tRNA and the P site and the last amino acid in the chain. So the polypeptide is free. So here's the free polypeptide. This release factor has released this polypeptide from this tRNA. And everything just really disassembles at that point. And now that everyone goes their separate ways. So you might think that one messenger RNA comes out of the nucleus and then you get one protein, but that is not the case. A number of ribosomes translate a single messenger RNA molecule simultaneously. That's called a polyribosome. So these many ribosomes in a row are called a polyribosome. So basically, with one messenger RNA, this messenger RNA, it's being read by many ribosomes at the same time. And this is a micrograph of what that looks like. So one gene gets transcribed into messenger RNA, and that messenger RNA is used to make the same protein over and over again by many ribosomes. The polypeptide chain also undergoes modification after translation. So those are called...
just to clarify, so all those ribosomes are making the same polypeptide?
Yes. Yes, these are all making the same one, exactly the same one. Yeah, that's a good question. Because they're reading the same messenger RNA.
Okay, start to end, each mRNA codes for a single polypeptide.
This particular messenger RNA, yes. Yes, it does. Yeah. A very good
sometimes
question.
They code for more, right? Or no?
Well, the pre-mRNA can be spliced in different ways. So the pre-mRNA can code for more than one polypeptide. Yes, that's a very good question. Yeah, it depends how that pre-mRNA is spliced and put back together. So there are different ways of doing that. So it can be spliced together with different exons. Does that make
thanks.
sense? Yeah. Yeah, thanks for asking that question. All right. So modification. So we know that proteins from macromolecules that have to be in a very particular shape to work, or they're not going to work at all. So they're folded into that particular shape, and there's some other modifications. So there's two different ribosomes in a cell. Some of them are free. They're just floating around free in the cytoplasm. Some of them are bound to an endoplasmic reticulum. So the free ribosomes, well, they'll initiate the synthesis of all proteins, but they won't all bind to the endoplasmic reticulum. Some of them will, some of them won't. The proteins that are destined for the endomembrane system, so that's the membrane system of the cell itself, or to be secreted, will be transported in the endoplasmic reticulum separate from the cytoplasm. Yeah, so there's signal peptides. The signal recognition particles bind to these signal peptides, and they enable translation ribosomes to bind to the ER. So that's another strange little peptide that's involved in this process. It looks complicated. It's not actually too complicated. This is a free ribosome here. It's already started a little bit of the translation. Here is the SRP, the signal peptide. There. And it binds to a receptor protein, which is here. That's kind of like docking at a wharf. You know, it binds there so that the remaining translation can occur inside the endoplasmic reticulum. And the reason is that this growing polypeptide will eventually be modified inside the endoplasmic reticulum and I should say, by the Golgi complex. So once the whole translation process is done, in this case, our peptide, polypeptide is now inside the endoplasmic reticulum, and everything else just cakes off. Messenger RNA. So this is important actually. This messenger RNA that you see here, it now gets digested in the cytoplasm of the cell. That's what happens to RNA. RNA doesn't stick around. Once it's transcribed, that's it for the RNA. That messenger RNA, boom, it gets digested into its nucleotides, and then they're available later on for new messenger RNAs to be made. And it's important at the moment, because what I've been hearing is that people are afraid of the COVID vaccine that is RNA. So the COVID vaccine, the Pfizer one and the Moderna one, they are both little tiny bits of messenger RNA from the virus itself that code for part of, not all of, the spike protein that's on the virus. So once that little bit of messenger RNA is delivered to the cell, which is done, interestingly, by a little lipid unit. Oh, what's it called? I forget now. A nanolipid something or other, I think, which was made right here, or at least developed right here in Richmond. So both of those vaccines have our nanolipids that are used to transport the messenger RNA to our cells. Once our cells get it, that messenger RNA, they go through this very process to make a little bit of the spike protein. And then that little bit of spike protein will be, through the process I'm about to describe, be delivered to the surface of your cell. And then your immune system will recognize it and kick in. I think people don't really understand the difference between messenger RNA and DNA. And there have been some things like, oh, it might change your DNA, but it can't change your DNA, because the messenger RNA gets digested, doesn't go anywhere. It doesn't go back into the nucleus. It can't. It just simply gets digested. So that's a bit of extra information I wanted to give you about the relevance of this to the COVID vaccine. So this is a table of all the different kinds of RNA. The ones that I would like you to remember for sure are messenger RNA. We know what that does. Transfer RNA, we know what that does. ribosomal RNA, we know what that does. There's this little precursor, a primary transcript. It helps with the splicing process. There's these, oh, this is what it stands for, I forgot. Small nuclear RNA. It plays a structural role in the spliceosomes. There's another one, the signal recognition particle. These are all RNA particles. So RNA is quite instrumental in this whole process. Then there's small nucleolar RNA, SNO RNA. They aid in the processing of pre-rRNA. And then there's these small interfering RNA and microRNA, and they're involved in regulation of gene expression. So we're not getting into those and what they're all doing, but I just want you to know of their existence. Prokaryote cells, interestingly, just a bit more, we're almost done here. They don't have a nucleus. They don't have a nuclear envelope. Awesome, Katie. So what happens is, here is your DNA in blue. That's your DNA. And all along this strand of DNA, there are RNA polymerases. They are making messenger RNA here. And that messenger RNA is being, here's one here that's blown up, is being immediately translated into a string of no, I want purple, of amino acids. So that doesn't happen in the eukaryote. The nuclear envelope separates transcription from translation. There's a lot of processing that occurs in the nucleus. So that may be one of the reasons why eukaryotes became multicellular and more complex. So I've just got a little more to go. I think I will continue, if you don't mind, to the end, just talk a little bit about mutations, and then the rest of the day will be all activity. But I did want to talk about mutations because, of course, a mutation in DNA can be detrimental in that it disrupts the codon sequences. Mutations are changes in the genetic material of a cell. A point mutation are changes in just one base pair of a gene. That's called a point mutation. It's different kinds of mutations. The change of a single nucleotide in DNA's template strand. So that can lead to the production of an abnormal protein. So in this case, here's a T, but whoops, there's a mutant. This is hemoglobin, a mutant, which instead of a T there has an A. So the mRNA, instead of having what it would have, has a U there. And well, that doesn't code for what it's supposed to, which is glutamine. It codes for valine. And that's the mutant for the sickle cell anemia condition that is caused by this mutant protein, which is hemoglobin. And that's just one mutation. Unfortunately, that mutation is in the middle of the codon. If it's at the end of the codon, it might not make that much difference. And I'll show you why right now while I'm on the topic. Sorry, don't get dizzy. I'm moving back. Don't look if it makes you dizzy. Going all the way back to the code. Bum-ba-da-bum. Ooh, ooh, not there, not there, not there, not there. There. So, for example, let's take a look here at serine. Oh no, even let's see, this is even a better one. Let's take a look at arginine here. So I'll blow that up, arginine. Its codons are CGU, CGC, CGA, and CGG. Those are 4 different codons that all code for arginine. Well, if there's a mutation in this one, it doesn't matter. It can be a U, C, A, or G. But if the middle one is mutated from a G to something else, well, that will affect the codon and its translation, of course. Okay, close your eyes again. We're going back. Does that make sense? So you're somewhat protected from mutations if those mutations occur at the end of the codon, the 3rd nucleotide in the codon sequence. So there can be base pair substitutions, base pair insertions, or deletions. A substitution is replacement that we just saw. It can cause either missense. So missense is what happens with sickle cell. So you still get a protein, but it doesn't make a lot of sense. Nonsense, you will not get a protein at all. So if the mutation is early on, for example, in the formation, then all of the codons might be out of whack, or you might have a stop codon. So here you've got stop codon that might happen very early on, so you don't even get the rest of the protein. Insertions are additions or losses of nucleotide pairs. They produce what is called a frame shift mutation, where now your codons are not in the same place they were before. So you've shifted everything. And that can be detrimental. Mutagens are the agents that cause mutations. They can occur in many stages. They can occur during replication, recombination. They can occur during repair, which occurs at replication when any errors are repaired. There are physical or chemical agents that can cause mutations. Let's see, UV radiation is one kind. That's why you should be careful in the sun. Some chemicals are mutagens, like nitrites can be mutagens. So there's a summary of it all. Transcription, which is happening in the nucleus. Here's your messenger RNA being formed. Processing, where some of your introns are snipped out. Your ribosomes coming together, your large and small subunits, ribosomal subunits, your tRNA ready, activated by the aminoacyl tRNA synthetase, and translation. Right, thanks for watching.